Sorry? We have 15 minutes. We're going to take a break at 9:30 thereabouts. We'll see how the discussion goes. But so I've got Vamsi and Alta. Anyone else? And Marnie. Okay, Vamsi.
First, I want to just thank both of you for really excellent presentations. That's very useful for our committee. Jacques, I have a question for you. I took some notes on some of the statistics that you reported from that one case series. I just want to make sure that I understand the numbers in that slide. In a series of 8 patients, you said that there were 2 in whom you could detect the donor mtDNA. Can you just elaborate? That means that you tested all 8 for evidence of the donor mtDNA. You detected it in 2, or you only looked in 2 individuals and you found it? And the second related question is, what was the tissue that you sampled? Did you look at the urine sediment, or did you look at a Buffy coat?
Yeah, good questions. So obviously, of those 17 babies born, the parents did not all consent to do the sampling, and the sampling was just buccal smears. Okay, it's just buccal, which I know is not perfect. And so only 8 babies were tested, and 2 of those were positive for donor mitochondrial DNA. There was other work published in prior series, the same patients. And we were able to sample amniocytes, and we found 3 out of 13 were positive. The others refused to be tested. And then we also tested those embryos that were not transferred because they developed abnormally. And we found that after 3 days from fertilization, 50% of them were positive for donor mitochondrial DNA. So there is a clear change over time. But then don't forget, this was 15, 20 years ago in the assays. I mean, it is very challenging to do this in fused cells, not necessarily on the buccal smears, but it's challenging to do this in early embryos. So we felt that that was probably an underestimation to find only 50% positive.
In these, of the 17 babies, how many of them are still alive, as far as you know?
No, everybody's there. Everybody's there.
And do you know what the current health?
Yeah, we've done a follow-up study. We started this process about 1.5 years ago, and we're completing the first series now. We need information on 1 patient. Still, once that is completed, we'll write it up, and hopefully it will be published in 3 to 6 months. As I said, the only thing I can say, it's reassuring. The data is reassuring.
Let me interject just on that. Can you share anything about what health status factors you assessed?
Yeah, so this is a first phase. And so the children, they're now vary in age from 12 to 17. So we assessed by survey methods how well they performed physiologically, clinically, in school, disclosures from the parents, etc. We never interacted with the children, only the parents, because disclosures which will be reported are very different from what we expected. So I think that the findings were reassuring, and we are suggesting, or we are proposing, a second phase study that would look at some of these children by looking at mitochondrial loads, for instance, and see what's going on. But these parents may not agree.
Alta, you're next.
Do you have a follow-up for George? First, thank you. I'm still catching up on the basic biology, so I wonder if you can just help me understand something I think everybody else might understand already. I'm trying to understand when you see these changes in the heteroplasmy, where you begin to get segregation between cells that now have high wild type versus high mutant load, right? Is this just a statistical phenomenon, and then you're seeing the 2 populations, and then you can select from one or the other? Or is there actually a competition factor in which some cells that have high mutant loads, or particular kinds of high mutant loads, are less fit as cells somehow, and they're being outcompeted by the ones with the normal load? And also in the kind of normal to normal, if you've got two different kinds of wild types coming from two different donors, is there any evidence that one type will dominate the other, or is it, again, just kind of pure statistics and segregation? I'm just kind of confused about what exactly is going on here.
We've tried very hard to answer that question, and we even actually asked Vamsi for help on this. And so if he can't figure it out, nobody can figure it out. And yeah, no. So there are clearly population dynamics, and we have a strong, I think, prejudice to believe that a cell with some kind of a defective load is going to be at a competitive disadvantage. We have done some mixing experiments to suggest that that's the case, but if you saw the performance of our different clones, it was somewhat unpredictable. So it can't be as simple as saying high heteroplasmy is at a competitive disadvantage. There must be some interesting interaction effect, because some of the clones, and not just in our lab, but in the Mayo study as well, seem to be remarkably persistent in their level of high heteroplasmy. So we've tried to manipulate that. We've tried to intervene in a way to change the bioenergetics so as to favor healthy as opposed to abnormal, but it's been difficult.
Can I ask just a follow-up? So just as a follow-up, you said that you were doing work also comparing the nuclear transfer process, and assuming that there's some amount of mitochondrial carryover from the cell that's providing the nucleus to the recipient cell, in those experiments, are you seeing any pattern developing in terms of which sets of mitochondria wind up dominating in the ultimate, kind of blastocyst that you're developing?
We have not done those experiments, and in retrospect, they would be interesting. And I would say that there are a few labs that are in a position to do that in the right context of human NT, such as Dieter Egley's lab and Shukrat Matalapov's lab and some of the European labs.
Thank you.
Marnie, and then I've got Keith and Jonathan and Alan.
All right, so unfortunately I do have 3 questions, so here they are, and you can choose which ones you want to answer. One, it's come up, this idea about different forms of mitochondrial replacement therapy, and some of the forms that have been recently introduced are polar body. And one of the questions as a geneticist that I have, and I don't know if it's known or not, is how would that potentially alter the genome or epigenome if you were to take something that hasn't completed meiosis and use that, such as the polar body 1? Is that potentially more problematic? Maybe not in terms of does it fertilize or not, but could it potentially have more epigenetic problems because it hasn't completed the process? That's question 1. I could stop if you want, but the other questions were, number 2, we've really been trying to consider what are the epigenetic alterations that we should be considering, and is there any knowledge in general? So for example, as a geneticist, we know ICSI itself has some percentage, that's not 0, of having methylation defects. And so are there considerations that you can guide us to think about the epigenetic changes that we might be introducing, by this methodology? Really, the nuclear genomic epigenetic changes is what we're interested in, and is one of these a better technique than the others when you compare polar body or chromosome spindle or pronuclear? That's number 2. And then number 3 is probably more for Jacques, but you are doing long-term follow-up, and we're really curious if you could maybe pontificate about what are the challenges or possibilities to do it systematically in America. So those are the 3 questions.
You want to take the polar body one?
The thing is, I had 2 slides on the polar body, so maybe this gives me an opportunity to answer that. That was work done in mice, and very well done in China, and published last year. They chose a mouse strain that has very nice polar bodies. It doesn't say that in the paper, nor have I seen heard comments like that. But that was done this way because Wakayama in '97 did the same thing to show that you can actually get offspring from just transferring first and second polar bodies to enucleated eggs or zygotes. And so it was an obvious approach. Normally, in most mouse strains, the polar bodies don't look good. Now, those people who work with mouse eggs and zygotes will agree, I think, with me on this. The polar bodies die very quickly. This is not a cell that's supposed to survive. I refer to them as kamikaze cells. And so, thinking that that would work in the human, yes, of course it would. It will give you a baby. The question is efficiency. Efficiency is important. And so all those three methods that you have listed and that you are debating will give you babies using any of those approaches. But the question is which one will be most efficient, because that's a safety aspect. And so if you look at polar bodies in the human, they don't look like anything like this mouse strain. They are on the way out the moment they are extruded. You can sample them for DNA analysis, and that's pretty reliable. But you have to do the sampling fast. If you wait several hours, the cells are really starting to fall apart, and I had some slides showing you how difficult it is to say whether that's one polar body or a fragment of a polar body. And so these polar bodies fall apart or they just degenerate. And that happens in about 25% of all polar bodies. So you're already diluting your outcomes by about 25%. You've lost that efficiency. And the efficiency rate may be, the loss in efficiency may be even higher, because some of these polar bodies will have lost their function. They're really not good cells. So I don't think this is a great approach, but it will give you babies if you go this way. So you have to choose between the other two or do them both.
Okay, so there was a bunch of things buried in Marnie's question. So you answered one part of one of those questions. George, do you want to weigh in on that? And I want to come back to the long-term follow-up question.
I might turn to the epigenome question that you asked. And I have to say, I think this is among the most active areas of exploration currently in the stem cell world. We're getting a much deeper understanding of the way particular culture conditions define the nature of methylation across the genome, for instance. There's been a renewed interest in going back to the human embryo and doing new derivations of the so-called naive human embryonic stem cells, which turn out to have a very different epigenome than the ones that Jamie Thompson and the rest of us have been culturing for the last 17 years. It's a different type of cell, and it performs differently. It's at a different stage of development, and it reflects a very different native equivalent within the embryo. So I think that the knowledge base is still evolving dramatically, but there is still a distinction between measuring function and measuring a molecular state of a cell. And where I can speak to this is the very extensive comparisons of reprogrammed cells via the Yamanaka factors generating iPS cells and reprogramming via somatic cell nuclear transfer. There's been a lot of hue and cry about the distinctions. In the ideal world, the iPS cell in its best embodiment is equivalent of a fertilized embryo-derived ES cell. But in practical applications, experimentally and all, you can define differences. You can define differences at the epigenome level, particularly in methylation and the persistence of somatic methyl marks and the establishment of aberrant new methyl marks that don't really exist. And yet those iPS cells in mice generate completely normal offspring. So there's there's a tolerance for a certain degree of variation in the epigenome at the functional level. And so I think I can't give you the right answer. At what level of molecular abnormality are we going to accept when, in fact, the best functional assessments we have say that things can develop normally? We're ultimately going to have to accept a certain amount of fuzziness on the molecular epigenome to move forward and actually get the data.
So Keith is on the list, but I want maybe Jacques can give us a very short answer about your thinking about the prospects for comprehensive long-term follow-up for children born from techniques like this.
Well, patients volunteer for these procedures and sign consents, and there has to be informed consent, and it will be a lengthy process, particularly for this procedure, as it was for cytoplasmic transfer. And in these consents, there could be a suggestion, a strong recommendation, that they should be tested prenatally. This happens in IVF and PGD all the time. And they sign off and they say yes. And then they refuse to do it 3 months later or 2 months later. Standard. So now you have to have patients in North America. This is not in England or in the Netherlands or Belgium. You have to have them sign off on long-term follow-ups. It's going to be very tricky. You can have them agree to it and they understand, but will they do it? I presume in mitochondrial disease, the commitment to that will be better than in any other that we have seen. Because in standard PGD, you look at the mutation and it's yes or no, except when there is an error in testing. It's just yes or no. So there the standard follow-up, doesn't apply. I presume these patients will be more motivated. But it is very difficult to make them commit to follow-ups, even when it's a few months.
Yeah. Thank you.
But who do you think should do the follow-up? So one is whether the patients will do it, but how could it even be done, is my question.
It's a very, very good point. So why did it take us so long to follow up?
Well, that's a good question, too. Right?
Yeah, well, these patients came from all over the place. So they were not in our care. I mean, we were an IVF clinic, and the IVF clinic, as it was that did the cytoplasmic work, changed because the different people in there, including myself, left, went to different programs, and now we had to get all together, and that took a while. And these patients just disappear and don't necessarily stay in touch, even if you contact them repeatedly. So it's not easy.
We need
Yeah, it's a
to use
pediatric
microphones when we're talking, but I think we get that there's a challenge. And I'm cognizant of the time. So I've got Keith, Jonathan, and Alan, then we're going to take a break.
Thank you very much. This has been a really helpful discussion in issues really crucially important to the work of the committee. And I think what comes across is this not only is MRT sort of technically complex, but there are a wide range of limitations in the analytics. What combination of techniques we need to consider to make it truly effective, PGS after MRT, alternatives, reprogramming, et cetera. And I just wanted to sort of double back to maybe pivot off the answer to the last question about the social policy and ethical challenges. So my question really is, from the standpoint of your expertise, what are the social policy and ethical challenges or major issues confronting the successful adoption of MRT in maybe a fertility clinic setting, at Boston Children's, or at other settings? Just sort of from your perspective, the follow-up is one of those social challenges. I wonder if you could sort of give us some sense of what some of the other social and ethical challenges that one might anticipate would be.
Maybe I'll just describe some of the experience that we've had through our bone marrow transplant program, which I think it kind of presages what might be happening here. I think we have a representative here who's been through this. I'm thinking of a family that was deeply Catholic and was unfortunate to have a very, very diseased first child, and they went through the struggle of deciding whether to have an IVF coupled to PGD for their second child, but to go further and actually select embryos that would be HLA compatible for a transplant for the first child. And when you start to add homozygosity and recessiveness and HLA and all, you get to a very small number of embryos, and it took them 5 cycles to get to a beautiful young child whose cord blood then was a life-saving transplant for the first. They went through deep personal review of their own religion and consulted with their priest and all, and they found a wide and diverse number of opinions and recommendations. But in the end, for that family, there was no quandary. There was no question what was the right decision. And now to see the kids, and they've now had a third child, it strikes me that we go through a lot of social concerns, but as a physician, I've always felt my responsibility is to the individual patient and to their family. So for me, I've gone through that on the PGD angle, and I would see a very similar kind of soul searching for MRT.
I think I like to add to that is who's going to pay for all this. If you look at in a regular egg donation cycle, it's $20,000 to $25,000. This is a cycle that involves 2 egg retrievals, not 1. So that will increase the expense. But also the checklist to find an appropriate egg donor is extensive and FDA regulated. So when you get suggestions like, I think it's an interesting suggestion from Doug Wallace yesterday to use haplogrouping, and you have to test for that. That's a considerable expense. Plus it dilutes your egg donor population. Egg donors get through all these tests and only less than 25% remain from the ones that you started testing. And so if you add haplogrouping to it, 1% remains. I mean, you just don't have that luxury. That luxury does not exist. So in haplogrouping, you would have to, you really have to probably look at what's the closest haplogroup if you want to apply it at all. And of course, some of the mitochondria experts say that, well, really, do you have to do that? There's no evidence that you have to. So, and anyhow, then there's the testing, the PGD testing, which in this case will be extensive. The extra freezing that you'll probably have to do. So it goes well above $50,000 per attempt. So how is that going to be taken care of?
So I'm going to read a couple of questions actually from Jonathan Kimmelman, who's, as you know, joining us from Berlin. So the first is to you, Dr. Cohen, and he says you mentioned that there were adverse events in the cytoplasmic transfer attempts that were reported to an IRB. So the implication being that the protocol was IRB reviewed and approved. So the question is whether there was some necessary obligation to undergo IRB review, or was that a voluntary matter that you sought prospective review by an IRB before going forward? Or were there legal mechanisms mandating that from your perspective? So that's one. And then the second is about, the second question even here is, would you say that if it's voluntary, is that a social norm among IVF clinics to seek out IRB review for innovative therapies like cytoplasmic transfer or others?
There are thousands of IRB proposals from IVF clinic each year. That's standard procedure, and I think it's legal, right? It's the legal way to do it. It's not a voluntary position. You can't do anything else. So if you do a procedure like PGD for mitochondrial disease testing, and it's done, I mean, we do it in the testing laboratory, so we are a reference lab. But if from the clinic point of view, dealing with the patient directly, that has to be an IRB protocol.
Well, it depends on where it's performed.
Well, exactly. And so there are exceptions. I presume there are exceptions, but certainly a series like an attempt like cytoplasmic transfer that requires in-depth full IRB review.
So at your attempts were, you understood them to be covered by federal regulations related to protection of human subjects?
Yeah, well, if you do that in IRB is covered by the FDA, right? That's the organization they report to. So, are supposed to. So, yes, it is.
Well,
yes, that is.
I'm asking really
Right.
how you understood it. I think that's the question rather than, I think we understand the scope of IRB requirements, but when you did that series at St. Barnabas, is that where they were
Right,
performed?
St. Barnabas Medical Center.
So the view was that you had to go through the IRB. It wasn't something you felt you wanted to
There was
do,
no
but
doubt
it was
about
required.
it. I mean, this is the first time I've heard a suggestion like that. Of course, you go through IRB review, and you do this every time, even when there's if you change the ICSI procedure a little bit, you get an IRB proposal. You go to IRB. I mean, that's standard procedure.
I'm going to do the second. Okay. And then this second question is also from Jonathan to you both. So yesterday, and I think Jacques, you were here, but George, you were not, so maybe we'll have to explain a little bit. But actually, Jeff Botkin asked what might constitute an appropriate preclinical package, so animal data, before launching into human investigations. So could you both enumerate the general categories of preclinical or in vitro studies that you'd want to see precede a launch of a clinical trial in humans? So the key outcomes you'd want the experimental evidence to rule out, roughly what the experiments might look like, sample size, outcomes, period of follow-up, species, etc.
Small question.
Now,
What
let me
your
say,
committee is supposed to do?
you can think about this for 10 seconds
mean,
while I say we're going to have
yeah.
another session after
Yeah.
the break that actually starts to take on some of these. So you are doing a good job of segueing us as well.
Yeah, I worried that question might come up, and I tried to immunize myself by saying I would greatly be invigorated by participating in that kind of discussion about how to define those. I would have my own suggestions about the degree of epigenetic analysis. I think it would have to be informed by a deeper retrospective look at how many analyses we've already done in the human IPS versus ES versus NTES. I tried to look at that quickly on the way down here, and there's actually a remarkably little amount of information and a very small number of papers. So I think we could define that prospectively, in part based on what data is in the literature. But I couldn't give you the statistics right here today.
Well, I'm confident that quite a lot of work has been done in the mouse regarding potential epigenetic effects of cytoplasmic transfer, nuclear transplantation, spindle transfer. Particularly a paper by Chang in 2009, this is from Keith Latham's group, and they must have spent years of work following up all these different permutations. They looked at different strains, they did intra-strain experiments, inter-strain experiments. And even in inbred strains, if they did the exchanges between one oocyte or one zygote from the same inbred strain, they found that this had minor effects on the offspring, or and these effects were transient, or there were no effects whatsoever. The effects were considerable when it was between two inbred strains, but I've explained that those are, I think, not relevant experiments. So I'm happy with those as precursors to this medical procedure. I think that was the most important set of experiments needed, except for proving, and it was done by, of course, Mitalipov and Dr. Turnbull's groups. They looked at minimal carryover from the mitochondria when you do these procedures, and I think that was accomplished very beautifully. So the only thing that's left is what I suggested, which is try to optimize it in a mouse model. Some of these more less sexy aspects of it, looking at membrane relaxins. Well, when do you do this, and how much do you use? When you do electrofusion, well, how can you optimize that? Don't always use the same settings. How can you change it? Same for Sendai virus extract. How do you change? How can you make that really safe? Because the toxicity studies have never been performed in basic studies. So I think that's what I would look at.
Can I just qualify that to some degree? I take a tremendous I take some comfort from extrapolating from model organisms. It's the basis of much of my research. But I also have a very healthy skepticism based on what we're learning about the differences between the mouse embryo, mouse embryonic stem cells, the human embryo, human embryonic stem cells. The experience of trying to extrapolate spindle transfer from the monkey to the human is sobering. So I do think we have to be careful about how much we conclude based on model organisms. It plays a role, but we have techniques and we should be paying as much attention to the analysis of actual human cells and the human process.
Thank you both. Well, we need to wrap up if we can. And Alan, yeah, you're last, and maybe Jacques can say what he wants to say in answer to your question.
Why did you stop, and why did you wait until now to do follow-up?
We stopped because all clinics in the United States received a letter from the FDA stating that they have jurisdiction and that you need an IND protocol to perform this. So we stopped because we needed a permit. We applied for the permit. When we stopped, this was in June 2001. We started applying for a permit after an FDA meeting on the subject in 2002. So in late 2002, we started applying, going through part of the protocol. It's called pre-IND. I think we can. I think I'm right in saying that we accomplished going through the pre-IND phase, and then we lost funding at the end of 2003. Because of the loss of general funding for the research lab, we couldn't continue this protocol. INDs really apply to pharmaceutical companies, and it'd be very hard to accomplish this just from a financial point of view because of the intensity of it for a regular clinic. I think we were on the way, and I think we probably would have gotten there. I hope we would have gotten there if we had another 1 or 2 years to interact with the FDA. They were very, I thought they were very positive and constructive. Your second, I think I answered the second part of your question, which is we waited because the group dispersed, and it was hard to stay in touch with the patients, and we have tried much harder now with this newly IRB-approved protocol, but even this has taken already 1.5 years to get in touch with 13 couples and to keep them committed.
Thank you both very, very much. It's been really instructive. I wish we could talk more, but we're really out of time. So thank you. We'll take a break until 10, come back, and we'll start with session 5.